Today we start with the O-Level Biology 5090 syllabus. Now this syllabus is being taught in most of the schools here in Pakistan. I am making this video on the January 2020, I'm making it in January 2021, but this is for the syllabus which is going to be followed in 2021, and the students who are taking the exams this year should be following this syllabus. Now, the first thing that we do whenever we start the chapter one, so this is basically going to be we're going to be talking about the chapter one, which is cell structure and organization. Now, cell structure and organization is the first chapter, and this is also relevant for the syllabus 0610, which is taught in IGCSE, but slightly the syllabus might be a little different. So students who are studying 0610 IGCSE need to go through that syllabus and see what are the syllabus points which they need to cover. Before we start any chapter, the most important thing is that you must have the syllabus with you. You must have a hard copy of the syllabus of the year in which you are taking the exam. Now this syllabus is for 2021. Now, the first chapter, and you must go through each point. And I still remember a student of mine who had written on each point, made notes on the syllabus, and put in the sticky notes and written on it on every point which he was not clear about or which he was had to ask me. And so he would go through point wise the syllabus with me and understand every point. So so you must go through, you must have this with you, and you must go through each point as we go along. When I teach you, I also keep the syllabus in front of me and I follow each point. Now, the first point that we're going to cover is the point A of the syllabus, which is examine. Now, examine under the microscope an animal cell, example from fresh liver, and a plant cell, example from Elodia, a moss or onion epidermis, or any suitable locally available material, using an appropriate temporary staining technique such as iodine or methylene blue. Now, the reason why we use a dye, iodine or methylene blue, is because whether it's an animal cell or a plant cell, it has no color. So in order to be able to see it, to visualize it, you need to color it. And the two dyes that they have asked you to use is iodine and methylene blue. And these are very good colors because they nearly stain everything which we want to see. But of course, for certain very particular parts of the cell which we want to see, we'd have to use some specialized techniques and specialized dyes for that. So the reason why we use dyes is you've got to understand is because nothing has a color. You can't see them. There are lots of bacteria in the air, but you can't see those bacteria. There are lots of virus in the air, but you can't see them. You can't see a molecule of oxygen in the air. So, in order to visualize these structures, we need to see them. We need to see them under the microscope. Of course, the microscope is just going to enlarge it, but we must be able to see them so they become prominent and you can easily identify. You can say, okay, well, this is the cell membrane, this is the cell wall, this is the nucleus, this has chromosomes in it. So in order to identify the different cell structures, we need to be staining them, and the two stains that we have used is iodine and methylene blue. Now, if you look at an animal and a plant cell, well, we have this a very basic diagram. Um, the basic diagram shows you that both of them have a cell membrane. So animal cell and plant cell both have a cell membrane, and please remember a cell membrane is partially permeable, which means we don't say semi-permeable. Semi means half. It means if you said semi-permeable means half the things can enter and half cannot enter. No, that's wrong. Maybe if you put 10 things, maybe 2 enter and 8 can't enter. So it's partially permeable. And of course, we'll understand it a little better as I go further into details. Then, of course, we come to the cell wall. So there is no cell wall in animal cells, but there is a cell wall in plant cells, and this cell wall is made up of cellulose. And why have I drawn this with a double line? The reason is that it is 500 times thicker than the cell membrane. The cell membrane is this gray line which I've drawn. And this cell wall is 500 times thicker than the cell membrane. I said thicker. And usually if I show you, if I am in class, I'm teaching you, I'll show you a register and I'll say this one page. The thickness of this one page is the cell membrane. And 500 such pages would be the cell wall. Then we have the tonoplast. The tonoplast is the vacuole membrane. You can also label this as vacuole. So the tonoplast is the vacuole membrane, and inside that would be the liquid, which is called the cell sap. But please, we do not write it on side. We should have done a label line to do it. So label lines are required. We never write on the diagram. I've made this mistake to make you all realize that we don't do this. Then the cell membrane are present in both animal and plant cell. Then cytoplasm is present in both. Cytoplasm is the gel-like or the liquidy part of each cell. So cytoplasm. Then we have mitochondria, which are the powerhouse, you say, or some of you say it is, they release. Now, important is that we use the word release energy. Please remember, energy cannot be created or destroyed. So mostly you write produce energy. Now that would be totally wrong. You cannot say produce energy. So the mitochondria is where the energy is going to be released. We go into a little more detail later on. So mitochondria are present in both an animal and plant cell because both are living and they both need energy to grow, to reproduce. Then we have a specialized area which is called the nucleus. This nucleus has a nuclear membrane. This nuclear membrane surrounds it and separates it from the rest of the cell. So it makes it a compartment. And that is why these are called organelles. Organelles are these tiny structures which have a specific function, like the nucleus has a specific function. It's an organelle. Mitochondria is an organelle. Chloroplast is an organelle. So the mitochondria, powerhouse, the nucleus, which contains a nuclear membrane, and inside we have the genetic information, which is on chromosomes. Please understand, chromosomes are the structure. And we say chromosomes are made up of DNA. Like I would say this chair is made of wood, or this table is made of wood. So chromosomes are made up of DNA. More details will follow. Then we have ribosomes. Ribosomes are the site of protein synthesis, where amino acids are put together and converted into proteins. So ribosomes are the site of protein synthesis. Now ribosomes are present in both animal and plant cells. Now coming to the differences are, the first difference is that there's the cell wall. The second difference is that there are chloroplasts present in some of the plant cells. Please understand, it's not present in all plant cells because remember, roots do not have any chloroplasts. There are many parts of a plant which does not have any chloroplasts. A red flower would not have any chloroplasts. A white flower would not have any chloroplasts. So chloroplasts. So chloroplasts will be present in some plant cells, but they're only present in plants. They're not present in animals. Then, of course, the cell wall is made up of cellulose here in plant. Then the third difference is that there is a structure known as centriole, and these are two rod-shaped structures at right angle to each other. And this is called the centriole, and these are only present in animal cells. So here are some of the similarities and differences. So if I ask you to compare an animal and a plant cell, I expect you to give me the similarities and the differences, things which are common to both and things which are only found in one. So similarities and differences is if I asked you in the question to compare animal and plant cell. Now this is a Venn diagram showing you the animal and plant cells. Now first a few things that I would like to add to this. Now the common things are DNA, ribosome, cytoplasm, cell membrane, cytoskeleton, mitochondria, Golgi apparatus, endoplasmic reticulum, nucleus, nucleolus. Now this is common to all. Plant cells have cell walls, they have chloroplasts, they have a central large vacuole, and they're usually a little sort of squarish or boxy in square, but this can be all kinds of shapes. And of course, we can add the centriole part to this. So they contain the centriole while there's no centriole in plant cells. So this is a Venn diagram, and you have to be understanding these because we come across this in the MCQs. Now, what we need to understand is that there's an artist view of an animal cell, which you can see here. And you can see how the structure has been very well colored. And of course, you can see it in a 3D dimension and you see how all the different parts. And you can see the labels. Please, everybody has a look at the label. The plasma membrane is the cell membrane. Then you see the centrioles. Then you see cytoplasm. Then you see there might be small temporary vacuoles. Then you see the mitochondria. And then this is the nucleus. And the rest of it ribosomes. Now the other structures which I don't want you to, so I'm not mentioning those. Endoplasmic reticulum, yes. Then now when you look at it under the microscope, you see a black and now this is a photomicrograph. Now a photomicrograph is what you see under the microscope when we take a picture of it. So that's why it's a black and white image, and especially if it's an electron microscope. So you can see the different parts and you see this part is the nucleus, which sort of colors a little differently. And then in some of these, you can see the mitochondria, which are these tiny structures. And of course, this has been labeled as the plasma membrane, which means this is the cell membrane. So this is what I want you to understand is how would these cells look on when you look at them under the microscope, whether it's a light microscope or a very highly magnified electron microscope image. Now, this is how a plant cell will look like. A plant cell will look like very differently. This is an artist view of it, and you can see all the nucleus, the nuclear pore, which is also the nuclear membrane is called the nuclear envelope because the nuclear membrane has holes in it, which are called nuclear pores. Then this also has ribosomes. Then the connections which connect two cells are called plasmodesmata. Then the plasma membrane, which has been labeled. And then, of course, there's the cell wall, which has been labeled. And inside the chloroplast, you have the thylakoid membrane and starch grains inside. And then you have this large vacuole. And you have mitochondria as usual, because mitochondria is present in both animal and plant cells. And then the cytoplasm has been labeled. And then there's the rough endoplasmic reticulum. Now, when you look at it under the microscope, this is how it looks like. Now, this part is the nucleus. So the nucleus you can see here, this is the large vacuole. You can see the vacuole membrane, you can see the plant cell wall, and you can see some chloroplasts in it as well. Now you must be able to clearly identify all these organelles on a photomicrograph. I wanted to pause the video here and then have a look at this and see how we will be able to identify these when this is given to you in an exam question. So an animal cell, you can see this is the nucleus. And here in this animal cell, this is the nucleus. And this is the part which is the nucleolus. The nucleus is the largest organelle. It is bigger than the mitochondria, it is bigger than the chloroplasts. So you can see the mitochondria here, which are these tiny structures. You can see the mitochondria here, which are the tiny structures. The chloroplasts are these structures, which are these big structures, but the nucleus is the largest structure. So this would be the nucleus. Now all you need to do is look up photomicrographs of animal and plant cells, put it in the Google search, go on to images and have a look at them. And that will make you feel comfortable in being able to identify animal and plant cells. Then we come to the last part of this chapter, which is the cell levels of organization. The level of organization is, first of all, we have organelles. Organelles are the tiniest of specific structures which perform a specific function, like mitochondria would be an organelle. A chloroplast would be an organelle. Then we come to the bigger than is a cell. Then many cells with the same structure and function would be tissue, so muscle tissue. Then many tissues together will make an organ. Like the heart is an organ. It has muscle tissue, nervous tissue, it has capillary networks. Then there is an organ system, and then finally there's an organism. And these are many of these examples which I have put in here: epithelial cell, epithelial tissue, epithelial, then skin, and this is called the integumentary system. Some of these may not be relevant to your syllabus, but like ciliated columnar epithelium, respiratory epithelium, lungs, and this is called the respiratory system, or the skeletal muscle, muscle tissue, then muscle, and then the muscular system. Then we have the red blood cell, and we have blood, which is the connective tissue, then heart, which is an organ, and then this is the circulatory system. So we need to go through these in different chapters. Of course, this organization has to be clearly understood. And as we do different chapters, we'll go through these different levels of organization. Now, as per the syllabus, we have to examine under the microscope plant cell, we say onion epidermis. And in animal cells, please, we do not use cheek cells anymore. So please do not mention cheek cells. This has been discontinued. So cheek cells is not to be talked about. We talk of liver cells. And liver, we can take chicken livers or we can take goat liver, and we can examine this under the microscope and we'll be able to see animal cells. Now, in the onion epidermis, we take an onion bulb, and with the help of a forceps, we remove a thin peel of the epidermis and we cut it up into a small piece. Then we take a slide and we place this small piece of onion epidermis on the slide, and then you add the methylene blue dye, and then you place a cover slip on top of it. And when you place the cover slip on top of it, now what happens is that this dye is slowly going to spread across, and then you place it on the stage of the microscope. You place it on the stage of the microscope, and then you examine it first under low power and then under high power. So the low power is a slightly lower magnification, which is 4x and 10x, and the high power is 40x. So you examine these under the two different magnifications to be able to see them more clearly, because just like when you zoom a picture, you see more details of it. But at times, of course, you do not see any more details because you magnify it to such an extent that more details, it just becomes blurred. So in order to see it, when you want to see it on the high power, you want to see more details of it. But then the light microscope has the maximum useful magnification is only x 1500. So even if you magnify it more, you're not going to see any more detail. It just gets blurred because the resolution is much lesser of a light microscope. You see, there are two words that use a lot: magnification and resolution. So magnification is just the ability to magnify. Resolution means to be able to see more details of it more clearly. This is another example from the plant system, or the plants which we have to study. We have to study different plants and animals. So one of the plants that here we are considering is a specialized cell, is a root hair cell. And then we have the root tissue, and then of course the organ is the root. And the system is the vascular bundles which make up the transport system. And this whole transport system results in a tree, which is an organism. So a specialized cell, then tissue, then organ, and then a system, and then an organism. So please relate this with plants and animals whenever you're studying the different cell, tissue, the levels of organization. Now going back to the microscope, you need to pause the video here and have a look at this. The first thing that we do is an onion is cut into quarters, and then we have one of the fleshy leaves is removed, and then we snapping the leaf backwards exposes the epidermis, and number four, a thin layer of the epidermis is peeled off. Number five, epidermis is placed on the slide and covered with 2 to 3 drops of distilled water, and the cover slip is lowered. Now cover slip is this little piece of glass which is made of plastic actually, and you place it on top of it. And number 6, then you add the stain on one side. So if you add the stain on this side, is put at one end of the slide because you're putting it after the cover slip. And then number 7, the stain is drawn over the specimen using a small piece of filter paper. So when you put the filter paper here, then the stain on this side is going to slowly be sucked in and is going to cover up the specimen. So please understand that what we are doing, we place the stain on one side, we place the stain on this side, and then on the other side we place this piece of paper here. So this is the paper here, the filter paper. So this is going to suck in the dye which was placed on this side. So slowly the dye is going to be taken in and then it's going to cover up the specimen. So this is how we do so how do we prepare a slide to be examined under the microscope. Now, before we end this chapter, I would like you to go through the syllabus again, and I would like you to revise it and see what have we missed out. We might have missed out something, but you need to go through it point wise. Now, the point here which I want to talk about is F and G. The first thing is the function of the cell membrane. The cell membrane controls the passage of substances into and out of the cell. So the fact that the cell membrane is partially permeable means that the cell membrane is going to prevent certain substances moving in and allow certain substances, so controls what is going to move in and out of the cell. Then the G part of the syllabus is function of the cell wall in maintaining the turgor or the turgidity within the cell. The cell wall, of course, is only found in plant cells, so the cell wall is going to prevent the cell from bursting. So when the water enters it, the cell wall is going to prevent that. So it's maintaining the turgor or turgidity is the word that we use. So the cell wall is important in maintaining this. Now all these which we have not talked about, we will be talking about in the different chapters. So the root hair cell is needed for absorption. The xylem vessels are needed for conduction and support because you see they are in the stem, the xylem provides a lot of support to the stem so that it can keep the plant upright. Similarly, the red blood cells have a very important function to play because they transport the oxygen from the lungs to your body cells. So these of course will be covered in the chapters that we go along. And as you finish this chapter and you read through the book, then you need to come back to this syllabus and see that you have understood each and every point. And then you should do the past paper questions from the paper 1, which is the MCQ, and the paper 2 questions also, which are on this chapter, so that you know how much you actually know the chapter. So just reading the chapter and understanding is not the main issue. The main issue is being able to do the questions which come in the MCQs and the different questions which come in the paper 2. This finishes this chapter, and I want everybody reading through the chapter from the book and then going through this video again and then seeing how much of the chapter you've understood. Thank you very much.